Okay. So last time we did all that, which I showed you right now. We did got into we got the expression table, and then we did a few of the techniques. We used some of our, important concepts such as log transformation, quantile normalization, and, other downstream analysis techniques such as PCA. So after that, let us just review what we covered. So if we transform our values into log scale, we will see a much more normal bar like. So this is the actual data, the first graph on the left. This is how your, a sample would actually look like. And you can see that a lot of samples are having very high expression, and there are also a lot of samples in down below which has low expression. But it is really difficult to work with this data, because this data is non-linear, and it's not even, it has a lot of, you know variability, which does not make sense. To make it more linear and make it more scalable, we do log transformation. So, we the first example, you saw fpkm values of a non-normal distribution, and then when we scale it, we do find that the means are somehow aligning. If you see the box plot, and if you see the line that is passing, that you will see that the means are still, at a level where you can have some sort of comparison. So now it is easier to compare these values based on the normalization. So let's see how this transformation affects the distribution of just, if I say one sample. So there are different distributions which we looked upon, and we also, after doing logarithmic, normalization, we learned about quantile normalization. So here you can see the box plot after doing quantile normalization, how the means are now much better aligned. So quantile normalization arranges expression value in similar order between samples and align their means. So you're seeing the alignment of means here. So you might want to do this for only one portion of the data, and we chose a threshold of 5. So why we chose a threshold of 5? Because many of the data which has, normal gene expression, which you might not be interested in, because while collecting, some data from your data from the sample at a particular state, many genes are functioning normally. So those genes which are functioning normally in both the samples are of not of much use to us, because there is no comparison there, because they are those genes are meant for normal functions. So we do quantile normalization to normalize that sort of data, so that we can focus on the data which is more relevant to us, to those genes which are differentially expressed, which has different expression. So we that's our uh, uh, that's our goal. So we learned that, and then we did pca. So before describing these what we got out of pca, and we got a nice scatter plot. But let me explain pca to you in a little bit more detail, because I think it's first one of the first machine learning methods that we are going to cover, which we are covering. So pca is a method for compressing a lot of data into representations that capture the essence of the original data. So what I'm trying to explain here is that we are trying to compress a lot of data without losing out the essence of the original data. Means that we are using this compression technique, but we are trying to keep the meaningful information from the original data. So pca compresses expression data from thousands of genes into single dot on a graph. So from thousands of genes from for a sample, it is showing you one dot in on a graph. So one dot per sample being analyzed. So how does it do this? So how PCA does it? So PCA does not select some characteristics and discard the other. It's nothing like that. Instead, what PCA does is that it construct a new variable. So it construct this new thing based on the whole transcriptomics data set, which provides a valid and accurate summary of variables being studied. So it looks into the whole data. It will look into your whole data. So essentially, each dot here represents number of reads multiplied by the influence of each corresponding genes on the overall variation in expression. So I think this is quite confusing, but what I'm trying to tell you is that these dots are coming after learning about the variation amongst variation in the data, and then PCA is trying to make meaning out of it, that these are the important samples, these are samples, and these are the genes. We can plot both and understand the variation which it extracts out of this big data. So talking about the nature of these components, so these components PC1 and PC2 which you see here are principal components. So these are the components that are derived out of that big data. So the components are characterized are on the basis of variation that exist in the expression levels. Sorry about that. So as a result of principal component analysis, we will get a table of values for each principal component with each object or sample in our data set. So you get, these sort of values, and column columns will also have a percentage. Here you can see in the columns, you'll see a percentage. That is the, amount of data this principal component has captured. So PC1, PC2, PC3. So there are different, components that capture the whole data. So we usually look into PC1 and PC2 and plot it, and also PC3 and plot it, because the PC1, PC2, PC3 captures the most of the data. So we did this in the last class, and we separated out based on, and we can say that we can visually see this, right? You can visualize your data by assigning coordinates to each sample based on principal components that best explains the variance of our data. Now we can visualize the samples on this scatter plot using PCA. So we are, you can see a separation here, that this class, the ER positive and the TN triple negative, can be separated into two classes. So that is something that PCA gives us. So we did what? We did quite a few things. We did build a pipeline. We got a table of expression, and then we did some normalization and logarithmic and quantile normalization, which is possible through the platform using the utility section. You saw that. Maybe I can just give you a demonstration later as well. Repeat that, but it's pretty simple to reach out till here. And we also did PCA using that data, and we found out that after doing PCA, we just can see that, okay, these are separated into two classes. So this is what we did so far. So this is now we are going into a transcriptomics 2. So Transcriptomics 2 is the second course in Transcriptomics series. In this course, we explain several statistical methods that can be used to find significant patterns in gene expression as well as their interpretation. So here we are, understanding a lot of statistics that are involved in with our data, and how we can compare, different data sets and make the meaning out of it, because that's the purpose of learning transcriptomics as well. So in today's workshop, we will be covering those things. So although we were thinking of doing, sort of, screen sharing, I think now if I can pause here a little bit and, ask whosoever wants to do screen sharing, I think Dr. Shinjay here wanted to do screen sharing, so we can do that and then return to the next segment, which is transcriptomics 2, and we will have a small overview with that as well. So Beepsha, can you help here with screen sharing?
Yeah, definitely. Dr. Shinde, you can unmute yourself and we can start here.
Okay, ma'am.
Yes. So you would see that in the Zoom meeting, you would see that besides invite manage participants, there is a share button, which is in green within your Zoom.
Yeah, can you see the green button? If you can press on that, I think it will start.
Yes. Once you press on that, you will be given different options like desktop, whiteboard, and all. You can just choose your desktop and click on the share button that is there below. Once you do it, we'll be able to see your screen.
In share option.
Yes, click on the share option. Are you getting desktop?
No.
No, once you click on it, there will be a pop-up and you will get desktop.
A screen option is there.
Okay. Are you getting any other options than screen?
Photo, document, box, Dropbox, Google Drive.
Okay, just go with your screen and let's see if it's showing your screen.
One more. I have opened it on mobile.
Oh, if you're opening it on mobile, if you have your laptop, that would be the best way to do it. Do you have your laptop ready? At the side, you can join the same Zoom meeting using the same link.
I have to just type same link on the laptop.
Yeah, just type for this Zoom link, there would be after that J, zoom.us/J, there would be a meeting ID. So click on the Zoom in your laptop and just enter that ID. That
Okay.
5, yeah, I will just repeat it for you again. It's 50167102 for today's ID and this meeting. And this goes to everybody. If you're using the app, sometimes they will ask you for a meeting ID and you cannot directly join via the link we send to you. So just use the number that appears after the link, which is after the J//. So that is the number which is always the meeting ID for any of these meetings we have here or any other Zoom meeting you might be having. And that is what you need to sign in.
I'm not getting the link right here.
Okay, let me write it to you in the chat box once more so that you just get that link here. Just give me 1 minute. I'll just get it for you. The link. This is the Zoom link for today's meeting. And
So let me go back to the presentation meanwhile, and once it is there in the laptop, I think we will be able to do it. Deepsha?
Yeah, please share your screen.
So as I was telling you guys that today we are going to start transcriptomics 2 a little bit. So transcriptomics 2 builds upon transcriptomics 1. So where we learned about processing of high throughput sequencing data. So here we will be introducing you to t-test, its history, the nuts and bolts of the formula, and give you some examples how statistics can be used for differential expression data. So after doing everything so far, after performing PCA and exploring the data in full, and seeing that our conditions are represented by samples we have, we can use hypothesis testing to select a set of genes that is most representative of the variation between conditions. Do you get it what I'm trying to say? So let me repeat that. So our conditions are what? Our conditions represents our samples. We can use, what we will be using is hypothesis testing to select set of genes. So we will select few genes using our analysis, and we want to believe that those genes represents the variation, that is there in those conditions. So if the experimental design is simple, or we just get a couple of conditions like disease versus control, we can use standard differential gene expression methods to select differentially expressed genes. So what we are trying next is, finding genes that are differentially expressed and then connecting it to the phenotypes. Means what is the, a biological question that we want to understand. So that is the phenotype. So if the design is more complex, we use another technique called ANOVA or factor regression analysis for multiple hypothesis testing. So these methods are commonly used, but they tend to, one of the disadvantage of these methods are that they tend to ignore variation that data has, because they are focused on a hypothesis and not learning from the data itself. So we will talk about those later, but first we have to understand what is, these concepts are. So introduction to transcriptomics 2 is something that we will be covering today, and then we will be doing hypothesis testing. So hypothesis testing, the concepts related to analysis of the differences between means of sample groups. So we saw the means, we have means from different groups. So we want to compare those. So how do we do that? We will be seeing that. And then differential expression analysis, which involves advanced methods for normalization, analysis, and interpretation of differentially expressed genes. And then we will be talking about factor regression analysis and other technical terms in transcriptomics too. So this is one of the these are the objectives that we have to reach. So talking about t-test, today we wanted to talk about something that is helpful for comparing data sets. So first, let's discuss how we can use statistical test to analyze a group of samples that might have considerable variation inside each group. So we are looking for variation within those groups, and we want to analyze them, analyze the data using some statistical test. To learn about this approach, we have to remember William Gossett. So William Gossett, as you see, was a statistician employed by a beer company back in the 19th century. So this is Guinness beer. So William Gossett was working with this Guinness beer brewing company, and he earned a degree in chemistry at Oxford and joined the firm in 1899. So his work for Guinness led him to investigate the validity of the results obtained from small samples. So previous theory was that he did it on a sort of large sample, but we will come to that in our next slides. So if you can see the the publication here. So he took a leave of absence to spend between 1906 to 1907 studying under Carl Pearson at University College London. So his publication in Pearson's journal Biometrica were signed student, not because a Guinness company policy for forbidding publication, as it is often said, but precisely because a company wished to keep secret the fact that they are gaining an industrial advantage for employing statistician. So Gossett's most important result is also known as student t-test distribution, which was published in 1908, as you see in this publication. So we wanted to talk about signal versus noise, because in our case, in our RNA-seq expression data, we get a lot of noise. But what we want to gain out of that expression data are the signals. So we are looking at the signals. So what is important for us in this hypothesis testing segment, or to understand t-test, is that we will be looking to derive signal. So a t-test is a type of inferential statistics used to determine if there is a significant difference between means of two groups, which I said already, which may be related in certain features. So we want to also find out the correlation, that how two groups are different, and maybe some features in that groups are related. So a t-test is used as a hypothesis testing tool, which allows testing of an assumption applicable to a population, because we are learning about samples here. So to select features that fulfill the criteria, we use a p-value. So you might have heard about this p-value. So the p-value is something that is based on the distribution of all values in our sample. So how we calculate p-value? The distribution helps select the observation that shows differences between two groups with low probability of an error. So we calculate all this using the t-test formula, which I will be discussing next, but I think we have a chat. Okay, so thank you for sharing this. So okay, so we want to understand by example, as I have been mentioning, and we wanted to discuss the hypothesis concept of hypothesis testing, and that too when we are comparing different conditions. So we saw in our example that there were two different conditions in our data set. One was ER positive, and one was triple negative. So those two conditions, and how we want to see what how they are different from each other, and how this test could help us doing that. So in statistics, t-tests are a type of hypothesis test that allows you to compare means, and they are called t-test because each t-test boils your sample data down to one number, the t-value. So for example, we can use a t-test to compare the means of two unrelated groups of samples. Okay, so as I was describing you the the data set. So estrogen receptor positive or ER positive and triple negative. So estrogen receptor positive breast cancer is identified by the presence of estrogen receptor on the cell membrane. If you can see this image, that you see that as estrogen receptors which sits on the cell membrane, uh, these receptors, when estrogen is available, comes and binds to estrogen, and then the cell proliferation is starts. So, and the triple negative one, the triple negative breast cancer has none of these three common receptors on this membrane. So what is happening in triple negative, and what is the difference in this, is that as a result, one is more treatable while other is more aggressive. So how one is more treatable and other is more aggressive? I think you should ask me this question. So this the answer is because we have these receptors on the on the surface. So what one strategy could be is to block these receptors by using an inhibitor. That is one way of treating that. But when you have no receptors at all, then there is nothing that you can do about it. The strategy would be completely different. So the other triple negative cancer would be more aggressive. So how do we know the conditions are different? So from the previous PC analysis, we know that the conditions are different, right? The difference is based on all of the genes, and all of the genes will have different profiles. So our goal now is to find the gene that are different as opposed to the ones that are not. Genes and observations. So differentially expressed genes are the ones we can identify to have clear differences in expression level between two groups. So here, if you can see, one gene is highly expressed in ER condition, and that gene here in the second image has no expression in another sample. So that's how you can differentiate that one gene which has in all of the samples, one gene has very high in ER high expression, and in some other it has a low expression, and in triple negative you saw otherwise. So we are looking at, here you can see that, if you can see, we are looking to find something like this from our data. What can a t-test be used for? So let's say that we have 100 mice, uh, 50 ER positive and 50 triple negative. Okay, so we want to find out whether a gene expressed in ER positive is significantly different from that of triple negative group. So we know that in the case of one gene, uh, but we want to find others that might be upregulated in one group and downregulated in other, or vice versa. Here is where we can utilize the t-test. So in each group, we have a number of mice, and each mice will have thousands of genes expressed, right? These are the observations that we want to test. For each gene, we test the group of samples for differences. So here, some variations between individual mice is expected because different expression. So if we would like to measure the difference between two conditions, we assume that our null hypothesis to be tested is difference between groups, and we suspect that they are indeed different. So we are thinking that they are different. So a test of hypothesis will reveal whether the significance, the difference is significant or not. So what we are trying to test here, that our hypothesis, we are thinking that condition A is different from condition B, and we want to prove that hypothesis. So what the purpose would be to, whether we can do it or not using the test. So in this case, we have two independent group of samples and a unpaired t-test, which can be used to determine whether the means are different from each gene. So let's look at the individual components of this formula and explain the variables. So for this formula, 1 and 2 represents two groups to compare, and x1 and x2 represents the means of group 1 and group 2. N1 and N2 represents the size of group 1 and group 2. And S is an estimator of the common variance of two samples. So this is a test that you can actually do on your Excel sheet. So there is a way of doing it, and that has been shown in transcriptomics too. The t-test statistics represents significance of difference between two groups. So we want to see that statistic, that what is the significance between two groups, and also, sort of, which gives us an idea of the fold change. So here, if you see N1 and N2, so the level of significance or p-value corresponds to the risk indicated by the t-test table for our calculated t-value. It should be noted that this t-test can be used only when two group of samples being compared follow normal distribution with equal variance. So that is one of the limitation of t-tests. So the p-value is often used often and represents an unlikely observation to happen by chance. If the p-value is higher, then the probability of that happening by chance is also higher. Since it is not by chance, we can be confident in the signal that observation displays. So this value shows the significance of the test. So the p-value that lies within this zone are the yeah. So if we see here this gene, I mean, if we talk about this one gene, so this ENSG gene listed at the top of the bar chart codes for this gene codes for mitochondrial enzyme ornithine aminotransferase. So ornithine aminotransferase is the enzyme, if I remember correctly. So this is a key enzyme in the pathway, which converts arginine and ornithine into important neurotransmitters like glutamate and GABA. So it is a pretty important, mitochondrial enzyme. So this is a key enzyme in the pathway which converts arginine and ornithine into neurotransmitters such as glutamate and GABA. So the y-axis on this bar chart shows the level of expression, where the bar represents an expression profile of a given gene or isoform. So you can see that the bar here is showing the level of expression. So differentially expressed genes are the ones we can identify to have a clear difference in expression level between two groups. So that is pretty much clear to everyone here, that some genes would be highly expressed in one condition and will have a low expression in another condition, and vice versa. So that's how we differentiate. So here you can see that gene has a higher expression level in triple negative group compared to ER positive group. So what do we consider while looking at differential gene expression is that there are thousands of genes expressed in a typical sample, which I talked about earlier also, and samples in each group has significant variance. So the variation is there between individual samples, but we have to understand some of them come from technical due to technical issues. We have to remove those before we do differential gene expression. Another important aspect is determining a threshold, which we decided based on the quantile. During the quantile normalization, we set a threshold of 5, and then we have to understand and then there is a difference that we have observed. So it's hard to know what would be a significant enough difference to say that these are truly differentially expressed genes. So that is something that is hit and trial method. But when you look into the output of DSIC, which is there in transcriptomics too, and also in our pipeline, which I will just show you, and we will end the presentation today, and then we will again discuss it in our next class on 3rd February. But one of the key aspects of when you run DSIC 2 is that you get a lot of information. So you get annotation information, you get visualization information, and you get a differentially expressed genes and a table of those things. So I think let us just go back and quickly do this pipeline today. So we will again do the same steps, and this time we will select differentially expressed genes. And then we did a table of expression. And once we get a table of expression, DSEC2 itself, does normalization, some sort of normalization log transformation, and then end. So here, if you can, download this dsec all file.zip, and then you will find the files that we were discussing here in the presentation. And this is again, if I go to the course here, this is well described in transcriptomics 2. So if you start taking this, and you can go through this so that you will understand also whatever we covered today. And in the next session, I think we will be covering factor regression analysis, and also some of the things that we covered today. So here you will have gene expression table, downstream analysis, which I talked about. So till here we covered in the last section also. Here we covered this. I was telling you about t-test in Microsoft Excel that you can do and see the result. And then differentially expressed pipeline and their algorithms and biological expression. This and factor regression analysis is with something that we will cover in the next class, and technical details and conclusion and summary as well. So as you see that we are going through our courses, and we are going in a systematic way so that it all makes sense. But this does make us to conclude today's session. So if you have any questions, happy to be happy to answer it now, and we can always answer the emails and answer on chat, and maybe answer in the next session when you are when you have practiced it a little bit. So as I was telling you that in the next session we will be reviewing factor regression analysis, and also we will be looking into some of the other machines. So we did PCA as well, and we will be doing factor regression analysis in the next class. And the class after that, we will be again repeating PCA with another tool, and we will do machine learning analysis, because I think after getting differentially expressed genes, next step is to use machine learning algorithms to understand about that data. So I think if you guys have any problem in understanding, once you go through these courses by your own, you will have a better understanding, and we will be connected. So thank you today for everyone for joining. And I think we will be doing this hands-on, which we tried with individual of you. So if you want a hands-on exercise with us, please write to us, and then we can have another meeting scheduled for you, and then we can practice as well. Bipsha, are you there? Can you do you have something to say?
Yeah. So for everybody who is facing any kind of problem, as Dr. Mohit already pointed out, please write to us. Like whenever you're having any kind of problem. So this goes for anyone having an issue with a pipeline, having an issue with the keywords that are there, the terms that are there, and we understand that this might be a little bit overwhelming for anyone who comes here for the first time, and that happened to each one of us. And that is how you learn, is like when you talk to us more and you say that this is the place where I'm having a trouble, so we can help you out in that regard. So one thing that we did not go over today, and if I would want to point out, is that we are seeing that a lot of you are doing the courses, doing the pipelines. So once you're doing the courses and pipelines, or just doing the courses, my recommendation would be to take a note down when you're going through the course. So if there is something that you do not understand, just note it down right there. All the things that Dr. Mohit talks about in this session is covered in the courses in a much elaborative manner, so you can always go back and refer, and you can see that what we are talking about in the sessions is already there in the course materials. But to relate all of them, having to take down notes or making pointers will help you to take the quizzes better and also ask us the right questions. So that is something that I had to share with everybody today. And we will be ending our session soon, and you all can actually email me at marketing@pine.bio. I know all of you have my email ID. So just if you want to set up a meeting with us for any time, you want to share your screen and go over your confusion, this is where I'm getting confused, I need help, so we can actually schedule those meetings with you over the next few weeks and then walk you through all of those problems. And definitely you can email to us, like any one of you, Pankaja, Priyasha, Priyanshu, anyone, if you're having any kind of problem about the terms, please email us the terms you're having problem with, so that we can send you the documentation that is required. And if you have any questions right now, please ask. Dr. Mohit is right here about this session. If you have any question, we can go over with it. And Dr. Shinde, have you been able to get your laptop right now? You can actually unmute and let us know. So we could actually do that with you if you want to go ahead right now. Because if not, then you can actually let us know a time you're comfortable in, and if we can also check our schedules, and then we can meet and take forward this session. So if you all do not have any more questions, I think that we can end today's session. I believe, Dr. Moore, do you have to add anything else?
No, Bipsha, thank you.
Okay, thank you so much. So please write to me, or you can text and let me know that this would be an ideal time, so we can send up that scheduled meeting, and you can let us know individually and personally your problem. So thank you so much for today's session. It was really nice to know about a lot of you who shared. I believe that in the upcoming sessions, when we have something like this, more of you would come up and share and talk about yourselves so that we can understand how you are doing. Yeah, and thank you, Soham. So if you are having any problem and you're still at Amity and IGE, please ask our campus ambassadors because they are present there and they can help you with any of these problems that you're having. Okay, so thank you so much, everybody.
Yeah, thank you. Bye bye.
Bye.